Now we are moving on to a new topic, that is enzymes. Okay, now enzymes, we know they are biological catalyst and they are going to catalyze a number of biological reactions in living organisms. Okay, so now if I am saying that enzymes are biological catalyst, that means they are going to speed up the rate of the reaction, and also they are responsible for some conversions that are taking place in our body. Like what conversions I'm talking about? Now the enzyme is going to help in digestion of food. Now I told you the food that we eat, it is in complex form, so when enzymes are going to perform their role out there, they are going to convert that complex food into simple food. Okay, so they play an important role in digestion of food, then absorption of certain molecules which produce energy, etc. Right, so enzymes, they are very important, or they play a very important role in our body. Now large number of chemical reactions takes place in our body. This is very much true. Without that we won't be able to carry out our daily functions. Now what is the requirement for enzymes to carry out their role? The pH should be 7.4. Okay, and the body temperature should be 37 degrees Celsius. Now, the reactions in our body are catalyzed by enzymes. So, I give you some example, right? Enzyme converting our complex food material into simple food material. Okay. Now, again, two examples I have given you. Insulin, which is secreted by pancreas, they are going to control the blood sugar level, right? And amylase, which is present in our saliva, it hydrolyzes starch. So basically hydrolyzes starch means it is going to react with water, and it is going to break that starch into further monosaccharide, right? So basically the same thing, it is explaining you that enzyme is going to convert a complex food or a complex substance into a simple one. Okay, now chemically enzymes are proteins, and every living cell has at least 1,000 different enzymes that are present. Now each enzyme is very specific in its action. For example, if an enzyme works on amide, it will work on ester or any other group. What does this mean? Means if you have enzyme, and for example if you take one group, okay, now take acid. So there is one enzyme which can work only on acid. So if you bring alcohol group or any other functional group, it will not have its effect. So each group has its own, और सबके लिए मतलब you can say as enzyme is very specific. Okay, so to break a particular compound, a specific enzyme is only needed. कोई दूसरा enzyme if you bring, then it is not going to work there. That is why it is said that enzyme is very specific in its action. Okay, now next we move on to the mechanism of enzyme catalysis. Okay, how the product is actually formed, how enzyme is going to work. Now action of enzyme is a lock and key mechanism. I think you have done this in your 10th standard, so it might be a revision for you all. Okay, so the action of enzyme is a lock and key mechanism. Each enzyme has an active site on it and a substrate of the shape of active site and fit into it. Okay, I'll explain you what is the meaning of this. Now once the substrate enters the active site, it is held in correct orientation to react and form product of the reaction. The product leaves the active site and enzyme is ready to act as catalyst again. Now formation of enzyme substrates complex has very low activation energy, and that is why it can catalyze about 10,000 substrate molecule in 1 second. Now this was the theory that is given under mechanism. Now I will actually explain you with the help of a diagram. Okay, now if you look at the first diagram, now this box with a V shape is called as the enzyme, and this V shape is called as the active site. Okay, so what they are saying is the substrate that is going to come and sit on this enzyme should be of the same size and shape as that of the active site. So if here it is triangular shape, you can see I have drawn substrate of triangular shape. If it would be curved, then I should have drawn substrate of curved shape. Okay, so only when the substrate is of the same size and shape as that of the active site, your lock and key method is possible. Fine. So now see, when the substrate comes and sit on the enzyme in the active site, it is called as the enzyme substrate complex, and this enzyme holds the substrate in a proper orientation, and this is called as the lock and key method. Okay, now here the reaction is going to take place and the product is formed. Okay, then the product will be given out. Now what they are saying here, formation of enzyme substrate complex has very low activation energy. Very low activation energy means it does not require more energy to activate itself. That is why as soon as the product is formed, it will immediately leave the active site. And they are saying like in 1 second, about it can, an enzyme can catalyze about 10,000 substrates. So imagine how how low amount or very less amount of energy is required for the product to leave its active site. Again I am telling you all, this is the enzyme, this is the active site. Okay, the shape of the substrate should be similar to that of the shape and size of the active site. Okay, only then it can fix it in the active site properly. Now you can see that the substrate has fixed itself in the active site, so this is called as the enzyme substrate complex, and this will show you the lock and key method. Now once the reaction takes place here between enzyme and the substrate, product will be formed, and once the product is formed, it requires very less energy to come out from the active site. So what they are saying is this one particular enzyme can actually catalyze 10,000 substrates in 1 second. So you can imagine how low amount of energy is required to activate this product. Okay, now this is one more example that I have shown here. You can see I have drawn an enzyme. Active side is V shape, but if you look at the size of the substrate, it is this rectangular in shape. So this substrate is not going to fix in the active site and the reaction is not going to take place. So the shape of the active site and that of the substrate should be same. Okay, and these are some of the examples of enzymes that have industrial applications. So the first one is manufacturing laundry detergents using proteases. So proteases is basically an enzyme which is used in manufacturing laundry detergents. Now laundry detergents, I hope you know, kapde dhone ka powder, right? Okay, now next, conversion of glucose to fructose by glucose isomerase. Next is manufacturing antibiotic by penicillin G acylase. Now I hope you all know what is the meaning of antibiotic. Okay, so manufacturing antibiotic by penicillin G acylase. So these are just examples of enzymes which are having industrial applications. I hope enzymes is also clear to you all. Thank you.